The first paper from our lab is finally out, and we want to share with you our story. We investigated the processing of a mouse coronavirus RNA during infection. Like many other COVID projects, this one started during the lockdown when we had a partial shutdown of the lab. Coronaviruses are positive strand RNA viruses featuring a 3' polyA tail. The length of the polyA tail changes inside the cell, but we know little about this process. In 2018, Eric Miska's lab showed that the uridylation of viral RNA polyA tail is a conserved defense mechanism. In collaboration, we found that TUT4 and TUT7 uridylate the mRNA but not the genomic RNA of the influenza virus, leading to a delay in viral replication. So we decided to investigate whether the same was happening with coronaviruses, and that is how this project began. In this study, we aim to understand if coronaviruses exhibit polyA terminal additions like uridylation or guanylation. We developed a splint ligation protocol compatible with nanopore sequencing to simultaneously determine polyA tail length and the presence of terminal modification. We discovered that terminal uridylation is a prevalent modification in coronavirus RNA. Approximately 9 to 10% of the risks have modifications, and polyA tails shorter than 22 nucleotides are more frequently uridylated than longer tails. Interestingly, the uridylation pattern changes during infection. The infecting genomic RNA or virion RNA has tails 66 nucleotides long, but as the infection progresses, the tails shorten and a prominent peak at 22 nucleotides emerge. Changes in polyA tail lengths coincide with a shift in the uridylation peak from approximately 60 to less than 22 nucleotides with an intermediate peak at 44 nucleotides. Coronaviruses have one genomic RNA and various subgenomic RNAs, which encode the structural proteins like the spike, the membrane protein, and the nucleocapsid. All RNA variants share the same 3' sequence, and we were able to differentiate them using our nanopore long reads. We found that the subgenomic RNAs drives the shortening of polyA tails and increased uridylation with a 22 nucleotide peak. The genomic RNA, however, shows symmetric and constant peaks of both polyA tail length and uridylated tails throughout the infection. Since TUT4 and TUT7 target tails shortened than 30 nucleotides, we examined the virus behavior in TUT4 and TUT7 deficient cells. We observed that infection progression was consistently accelerated in the mutant cells. Moreover, viral RNA load increased approximately 8-fold in TUT4 and TUT7 deficient cells, consistent with TUT4 and TUT7 promoting RNA decay. To understand what is happening at the molecular level, we analyzed polyA tail and uridylation profiles in control and mutant cells during infection. While control cells display polyA tail shortening and the emergence of uridylated tails at 22 nucleotides, this did not occur in TUT4 and TUT7 deficient cells. Instead, polyA tail profiles remained symmetric, and uridylation at 44 nucleotides became the most prominent. Examining the different viral RNA species, we observed that polyA tail profiles of the subgenomic RNAs were significantly affected upon TUT depletion, but not those of the genomic RNA. We propose that TUT4 and TUT7 promote short-tail uridylation of subgenomic RNAs, leading to transcript decay, and the reduced viral load then impacts the virus replication capacity. In conclusion, while TUTs only delay viral replication, they significantly affect RNA processing by blocking the transition to an unstable state. Inhibiting TUT's activity could help capture RNA intermediate state and facilitate our understanding of the machinery affecting the transition between different RNAs. With this, I want to congratulate Ankit Gupta, the lead author of this study, for all his hard work and dedication. And also I want to thank everyone in the lab and in the institute for their incredible work and support.